Hi everybody, welcome to the Countdown Mastitis Diagnostics webinar. I hope you're all managing to keep busy and stay safe at the moment with regards to COVID. It's been some pretty interesting and challenging times for us all, but really excited to be bringing you the webinar today. It's a good, nice social distancing activity that you can do. Before we begin the webinar, firstly, I just wanted to introduce myself. So my name is Steph Bullen and I'm the lead of Animal Health and Fertility at Dairy Australia. I know many of you, but for those of you I don't know, I'm a vet and joined the DA team back in October 2019. And I look after a number of well-known programs such as Countdown, In-Calf, Rearing Healthy Calves, Healthy Hooves, etc. So before we get into the webinar, I just wanted to run through a little bit of housekeeping. So for your information, the webinar is recorded and it will be made available to those who have registered and other participants after the webinar. So you'll see that you're all muted and that I've been turning your cameras off. The reason why we're doing that is just to make sure that we've got enough bandwidth for those of you that don't have the best internet connection. We will hold questions until the end of the webinar, but if you do think of any questions during the presentation, if you can just enter them in the chat box. So if you're on a laptop and you just hover your cursor down the bottom of your screen, you'll see the little chat icon there. And if you're on a smartphone, if you just tap the screen, you'll see 3 dots in the right-hand corner and you can then follow the navigation to the chat box. Please also just give me a shout via the chat if you're having any issues during the webinar. So it's my great pleasure to introduce our presenter today. Dr. Zoe Vogels is a dairy cattle vet from the vet group in Timboon, and she's been working there since she graduated from the University of Sydney in the year 2000. Zoe is a member of the Australian and New Zealand College of Veterinary Scientists in Epidemiology and Dairy Cattle Medicine, and she's got special interests in both calf health and milk quality, and she's been a member of the Countdown team for several years. During her involvement with the Countdown project, she's led the development and review of a number of tech notes and farm guidelines, and she was a deliverer of the Advisor Short course. She's also delivered numerous cups on, cups off courses directly to farmers and continues to do so. So most recently, Zoe rewrote the bacterial identification FAQ sheet, which sits in the Countdown tech notes. So many of you will be familiar with this document, but the rewrite has the purpose of the rewrite really was to include information around on-farm and in-clinic diagnostics, which weren't available at the time of the last writing of that document. And so the rewrite of that FAQ sheet really sort of forms the basis of this webinar. The FAQ fact sheet should be available in the coming weeks, and I'll send it around directly to all of those who have registered for the webinar, but it'll also be available on the Dairy Australia website. So without further ado, I'm going to hand over to Zoe.
No, I'm still muted. Alt A, hang on.
You're good, Zoe?
You've unmuted me. I tried to unmute myself, Steph, and I couldn't do it. So anyway, hello everybody. It's good to see so many people logging on, keen on milk quality. It could be that you're just desperate for some form of human interaction, but that's okay too. And a shout out if there's any school kids who are sitting in and this is their science lesson for the day. So we're going to talk about mastitis diagnostics. There's a few things that we want to cover. Oh, hang on. Steph, my screen has frozen. No, it hasn't. Here we go. So what we're going to cover today is we're going to talk about why we want to do mastitis diagnostics and rapid diagnostics in particular. We're going to talk about some of the systems that are available, how you might assess the different products that are available, how you might implement a rapid mastitis diagnostic system on farm or in one of your clinics, what are the limitations to these systems, and then briefly a little bit about PCR and antibiotic sensitivities. So why diagnose? And this is, it's pretty much something that I remember one of my colleagues saying to me, Monica, years ago. She said, only carry out a diagnostic test if the results are going to change your course of action. You don't just really do it for the fun of it. It's got to be something where it's going to change how you proceed. So why do we diagnose the cause of mastitis? I guess it's a vital part of a mastitis investigation. And this is the flow chart from Tech Note 13, which is how you approach a mastitis problem. And the second step is define the problem more specifically using milk culture results. So it's a really important part of a mastitis investigation, helps us with treatment and culling decisions. It helps us identify whether we've got an environmental or a cow-associated problem, and that helps us focus on prevention strategies. Historically, we do these mastitis cultures using individual cows, and often we'll use it in conjunction with a bulk milk or waste milk PCR. And I hope most people have seen an example such as this. This is the Complete 16 PCR test, which I think is the one that's most commonly being used at the moment. We don't often do PCRs on individual animals because it's a bit pricey, and we also use it when we're purchasing cattle. I guess the next question is, why do we want a rapid diagnostic system? Sending milk samples to the lab can be a long turnaround. It can be more expensive, and often the advice used to be, for farmers to save up, milk samples in the freezer and then send them when you had a whole lot. But it's a bit more expensive. Some clinics will set up their own labs. So this here in the photo is up at the Tablelands at Malanda, where Sam Rowe set up a lab, and Ryan Cockram has taken it on. But it is still a little bit complicated to set up a lab yourself. You still need to, identify the bacteria or grow them, identify them, do some enzymatic tests to work out which bugs you've got. And what I would say is I've just included a couple of relevant references on slides. So if you go, oh geez, I'd actually really like to set up my own lab, this is a reference that you could use. Why rapid diagnostic systems are being used is because they are easier. They're easier for more people to do, and they also allow you to identify an active infection, and this then allows you to do selective lactating cow treatment. So with regards to selective lactating cow treatment, this is something that's happening in North America, in Europe, in the UK, and is starting in Australia. So this is culturing on farm and not treating no growths. Some farms might also not treat mild or moderate clinical cases caused by coliforms or Staph aureus. And this, it results in less antibiotic treatment, which means less money spent on antibiotics and discarded milk, and also a lower risk of antibiotic residues. So, for all of you guys who are working at milk factories, this is probably one of your big things. Now, I guess with selective lactating cow treatment, it's something that not every dairy farmer has been exposed to, and it's something that they might have questions about. It's a bit of a new concept. So this slide here, it's not really something to spend lots of time on now, but I think it would be useful for everybody to have, just a little cheat sheet to refer to of what are the, what studies have been done, what are the pros, what are the cons, and I just included these from another presentation I've done, because you might be able to have them on a sheet in your car when you're talking with farmers. So just as an example, this top paper here, they looked at 473 cases of mastitis. Half were treated with intramammary antibiotics and half weren't. They saw a reduction in antibiotic use. So, they halved the antibiotic use, and they saw no difference in days to clinical cure, bacteriological cure, new infection risk, recurrence or removal from herd. So these are just some things that you can add to your little cheat sheet in your car. One thing I would say about selective lactating cow treatment is the not treating takes a significant change in mindset. So this is a quote: "Was there really no growth? Because there were lots of chunks." And it's not just farmers, it's also vets, I find, where being able to just like that shift in mindset that just because you see lots of chunks doesn't actually mean there's bacteria there. And I think it's really important for us all as milk quality advisors to be able to explain how clinical signs do not necessarily mean the presence of bacteria, which means you need a good understanding of how the immune system of the udder works and how the cow fights an intramammary infection. And here's just another reference. This is one that helped me kind of work it out in my head about how the udder works. So again, just a reference for you to to have a read of sometime. What rapid diagnostic systems are there? There's a few. The first one here is petrofilms. So these have come from the food safety field, and basically they're little films which you lift up, you put some milk on, or some diluted milk on, and then you flatten it down, and then you put it in the incubator, and 24 hours later you'll have some growth. So each cow gets 2 different films. So this one here is the gram-negative film, so you can see those little tiny dots, that's gram-negative growth. This other white film here film is the gram-positive growth, so all those little pink dots is gram-positive growth. When you have nothing growing, that's a no growth, obviously, and this here is a mixed growth, so there's growth on the gram-positive and also on the gram-negative side. So these are cheap, they save on shelf space, and I think they have a pretty long shelf life as well. The next system, they're called colorimetric systems, and basically this is where milk is added to little wells, and the bacteria is identified by what combination of colour changes are seen. So, for example, and this is the Mastitis test. That analysis is done by a little computery thing that works out which combination here tells you which bacteria are present. This is the speed mam colour, and that's actually where you physically have to look at the different colour changes and then work out which bacteria you have. At a very basic level, this is, I think it's Mastitis. So basically it's saying, looking at just gram-positive, gram-negative and no growth based on those colour combinations. So is it 2 pink tubes, a pink and a white tube, or 2 clear tubes? The next systems are the rapid mastitis plates, and the Minnesota easy culture plates. They've been around for the longest, and I hope people have either seen about them or heard about them. And basically they have biplates and triplates, and depending on how bacteria grow, they can work out what bugs they've got. So these are used like McConkey Agar and Focus and Factor Agar. The next type of rapid mastitis plate are chromogenic plates, and so with, you know, in that lab that they have up the tablelands, they look at changes in the agar. So is there hemolysis, is there pH changes? So rather than relying on changes in the agar, chromogenic plates rely on colour changes within the bacterial colonies themselves. So the agar's got these little things called chromogens, which has got a substrate and a chromophore attached. And when an enzyme specific to the bacteria you're looking for breaks that bond, the chromophore becomes insoluble, and that kind of precipitates and it becomes coloured. And some agars, they'll have more than one chromogen so that they can detect multiple bacteria. And there's lots of chromogenic plates out there. Once you kind of start looking for different products, they're being used more and more, also in human medicines as well. So for example, over here we've got, this is Agar Sense that comes out of Canada. And what I would say is, for some of these plates, not all of them are chromogenic agars. So that's a chromogenic agar there. This is Acumass that comes out of America. This is the checkup plates that come out of New Zealand. A bit of a disclaimer, we use the Acumass plates at work, and so they're the ones that I'm most familiar with. And as someone who really likes, well, I guess I'm what you'd call a visual learner. I like colours, I like drawing, I love photoshopping stuff. So these are right up my alley. And if I come across as a little biased, I apologise. It's just because I guess you could say I really like all the pretty colours. With the agar plate arrangements, there's a few different ones that you've got. So you've got your biplates, and they usually class things as either gram-positive or gram-negative. With your triplates, so up here are your different brands, but they'll have a gram-negative third. They'll have a third that looks for strep and strep-like organisms, which is otherwise known as SSLO. And then that last third will be either staph species, or it'll be just gram-positives, or it'll just be everything. And then you also have quad plates, and they always seem to have one quarter that is just blood agar, so it looks for everything. There'll be a quarter that's gram-negative, and then those last 2 quarters, they'll either be, there'll be one that's just Staph aureus, there'll be one that's strep, or it might be a gram-positive quarter. So there's a little bit more variation in what those two quarters are. There are some other benefits of the Agar plates, I think, compared to the other systems. One of them is that it allows assessment of the amount of growth. So you're not going to get this in the colorimetric system. So, for example, I find this really useful when I'm talking with farmers, say at a cups on, cups off course. So I can say, for example, in this plate, you can see, look, each of these dots is a bacterial colony, and you can see that this is a mild or moderate growth. Whereas this one here is a heavy growth of bacteria, and this one here, so this third over on the right, there's so many bacteria there that you can't even see individual colonies. And then you can say to them, well, how much milk is actually spread out on this plate? It's about 0.1 of a mil. So if 0.1 of a mil has got so many bacteria, that explains why just one drop of milk can be really infective. And I find this just a really valuable visual tool when we're talking about the spread of mastitis in infected milk at milking time. The other benefits is they do, because of how they're arranged. So what I should say is each of these thirds is designed to suppress the growth of the bacteria that they don't want to grow. And so it does allow you to identify mixed infections. So that's when there's two bacteria present. So these two plates are showing two different gram-positive infections each, and you wouldn't be able to pick this up on a Petrifilm, for example, because that's just gram-positive. So you'd say, oh, it's gram-positive, but you might miss that there's two bacteria present. The other thing is they allow good assessment of farmer, or put in bracket, or vet hygiene. So this is a photo from Lucy of some checkup plates, and you can see, hopefully, that there's some blue ones here, which I think are strep. There's some blue ones in this quarter and some pink ones as well. So that's, some coliforms. But 3 different types of bacteria. So you could say to this farmer, well, I guess there's 2 options. One is this cow is actually infected with 3 types of bacteria, or she's, it's been contaminated when you've collected the sample. So let's talk about your contamination technique. This picture here, I think, is such a corker. This is from a milk sample from just a couple of weeks ago down here. And I mean, there's so many different colours and so many different types of bacteria, just heaps. But what it did do is it helped me talk to the farm staff about collecting samples and how they might do it better to reduce the risk of contaminating samples next time. So they're just really good visual tools for farm staff when we're talking about collection of milk samples as well. So how do I choose a product? I guess the thing that's good to do is choose a product that is going to do what it says it does and has published research of its results. So for example, I kind of want some proof that ivermectin is going to stop COVID before I start using it willy-nilly. And in terms of published research, you want it to be compared to a recognised reference test. And generally this is the National Mastitis lab culture. So I'm not sure how many of people have even heard of NMC. It's the National Mastitis Council. It's kind of like a worldwide group of milk quality nerds. And they produce a lot of publications, one of them which is a laboratory handbook. So if you're keen on milk quality, milk microbiology, it's a really good investment. You can buy it online. But most people, that is kind of the reference go-to when they're doing lab culture. I should note that more labs are using what's called MALDI-TOF in addition to culture. So rather than using all the enzyme tests to identify bacteria, they sometimes using MALDI-TOF, which stands for Matrix Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry, which is why we say MALDI-TOF. And I will just put a little diagram that talks about what it actually is, because Steph mentioned that she'd had some questions in a previous webinar. So basically what they do is they get a colony. So pick a colony from a plate, you add it to a little crystalline matrix and put it on a plate. You hit it with a laser beam, which kind of ionizes and suspends the proteins in the air. And then they run it through an electron field generator that kind of accelerates the particles, and they separate based on their mass or their size, their charge per size. And then they hit this detector that kind of measures how long it's taken to get there. And then you end up with this little spectra of all the different proteins that are there. But then that spectra has to be compared with a library. So this is the catch. If your MALDI-TOF machine does not have a very good library, then you're not going to be able to identify the bacteria as well. So yeah, so this is just the kind of new stuff that's coming out. And yeah, I guess if you have any questions about it, maybe Google can be quite useful trying to find out more about it. So in terms of looking at the, you know, how to choose a product, you also need to look at the stats. Now, I know that stats is not for everybody, and reading papers is not for everybody, but you can't, well, new tests are going to appear and you need to be able to assess them yourself. Just because the President of the United States says, hey, you should try something because what have you got to lose, doesn't mean you should do it. And I just, you know, it's been a tough month or so, but geez, if we think we've got it bad, just imagine being Dr. Anthony Fauci, advisor to the President, and what would drive him to touch his face in the time of COVID. But with regards to the tests, there are some key measures. There are calculators on the net that will help you work them out. And if it's really not your cup of tea, this is where you phone a friend, find someone who does like it, say, hey, I found a paper, can you help me work out these stats because I just don't get them. I'll swap you some toilet paper or a loaf of sourdough. You know, there's something that you can give them in return for them to help you. And there's some key measures to look for. So with regards to stats, this is what we call a 2x2 table. So you have your new test and whether it's said something is positive or negative. And then we have the reference test. So for example, the NMC lab culture and whether that's positive or negative. And then we look at some key measures. So the first of them is sensitivity, and that's the ability of a test to identify infected animals correctly. So if you have a sensitivity of 80%, that says I will identify 80% of infected animals correctly. Specificity is the ability of a test to identify non-infected animals correctly. So you can say, oh, it's a negative test. So sorry, a specificity of 99% means that 99% of animals that are non-infected will be, that's what we'll say that they are. And then there's also the positive predictive value, and that is the likelihood that a positive test is actually correct. So if you say, help, this test says that this cow has staph, its positive predictive value is 80%, that means that 80% of all positive tests are actually correct. And then the negative predictive value is the likelihood that a negative test is correct. So those are some key measures when you're looking at and comparing tests. Another one is what we call the kappa coefficient. And this is like a measure of the strength of agreement between a new diagnostic test and the reference test. And it kind of controls for whatever correct agreement might be due to chance. So they have down here is just a list of all the different categories. So obviously if it's 0, it means that there's no strength, no agreement. If it's moderate, if it comes, you know, anywhere from 0.4 to 0.6, it's moderately, there's moderate agreement, and if it's more than 0.8, it's got very good agreement. So I guess just an example of how to use this kappa coefficient. There's some references right at the end of the webinar, but this one, so this was Griffion, and they compared 4 different rapid mastitis tests, and this was some techs and a vet's interpretation of the test versus lab culture of the sample. And this was interesting in that there was different levels of agreement for different categories of bacteria. So for gram-positive bacteria, there was poor to fair agreement, whereas for gram-negative bacteria, there was good agreement. So the kappa here was 0.7 versus 0.4 to 0.25 for the gram-positive bacteria. So that's, of all the tests, their agreement was 0.14 to 0.25. And then there was fair agreement to no growth for, so there was fair agreement for the no growth category. So I guess from this paper, I'd say, well, actually, it's not just how a test functions actually depends a little bit on which bacteria are there. This was another paper. This is Ferreira. They looked at 4 different systems. This paper, they had a trained tech. It was trained tech doing the collection of the samples and also interpreting the plates. And they had these products had moderate to very good agreement for classification into gram-positive and gram-negative categories. And they even had moderate to good agreement for classification into genus and species. This was looking at the Mastitis data, and this is the machine's interpretation versus lab culture. And this was classed into, like they classed the results into coliform, strep uterus, Staph aureus, and no growth categories. And they all had fair agreement ranging from 0.3 to 0.38 for the kappa. And then the next one was McDougall, which looked at the checkup chromogenic plates. And this was farmer interpretation versus lab culture. And the results were categorised according to their planned treatment. So there was Staph aureus and gram-negative groups and no growth and other groups. So the other included like your streps and coagulase negative staphs. So for Staph aureus and gram-negatives, they had moderate agreement, but there was fair agreement for no growth and other. So I guess what I'd take home from this is that if you have a really trained technician, like it's hard to compare these 2 papers because this is done by a trained technician and this is a farmer's interpretation on farm. So it's hard to compare. I mean, obviously this one hasn't performed as well as these ones, but the actual how the study's been run has been different. Now, what I would say is all of these studies have been done overseas, and hopefully we'll have some Australian research in the near future, which looks at Australian farms and can hopefully, I guess, compare them all on the same level. So using the same people doing the interpreting and the same samples. So how do I choose a product? You're probably thinking, hey Zoe, you haven't really helped me any yet, because all these studies are done differently. They've got different comparisons, different categorizations. So I've made up this graph and I hope it's useful. So if you have a product in front of you, your confidence, so up here, your confidence in its performance, if you have, the more peer-reviewed papers or papers showing high sensitivity and specificity and kappa you have, the higher your confidence in the test is. If you have increasing levels of bacterial identification, so up here you'd have gram-positive, here you might have genus, here you'd have down to species, increasing level of detail, you actually have decreasing confidence in how well it's performing. So you're more likely to have like a misidentification here than up here. And actually what McDougall said in his paper was it was actually, if you did try and look at, if they'd taken that level of agreement down, down to species and genus level, that agreement would have been worse. So I guess if you've got a farm that is just doing selective lactating cow treatment, a test that distinguishes between gram-positive and gram-negative may be all that you need. But, so you have real simple, you have a biplate and you say that is a gram-negative and this is a gram-positive. So really easy. But if you're someone who's wanting to use this as part of a mastitis investigation, so this is where you need to be able to categorise into cow-associated and environmental bacteria, that species and genus accuracy becomes more important. So for example, yes, you might be able to identify something into strep, but strep uterus and strep agar are distinctly different when you're categorising them into cow-associated and environmental bacteria. So if you're wanting to use these for mastitis investigations, you need a product that proves itself more in the species and genus accuracy. And this is just a slide, it's come from a different presentation that I put together, but just highlighting that, that this classification into environmental and cow-associated bacteria is important because how you approach them is different. So, for example, for environmental bacteria, it's about minimising contamination of teats with feces, mud and water. You might be really evaluating the hygiene of the storage and infusion of intramammary products. Whereas for cow-associated bacteria, it's a big focus on milking hygiene and teat health. Probably what I haven't put in there is ensuring good teat disinfectant coverage post-milking. The other thing, I guess, is when you're looking at a product is considering the implications of a false negative or a false positive test result. And that's, I guess that's the test performing in perfect circumstances. And it's also, part of this is also the potential misinterpretation of those tests. So if it's a test that's a bit complicated and is more likely to be misinterpreted, then you'll end up with a false negative or a false positive. So for example, if you're deciding whether to treat or not treat, how likely are you to treat a cow when the cure is unlikely, or treat them when an active infection or not treat them when an active infection is present? So for example, if you've got a cow that is, if you've got a farm that's an organic farm, and if they get treated with antibiotics and that means they're out of the vat for 6 months, if you've treated them when they didn't, or they shouldn't have been done, you've cost that farmer a lot of money. Or if you've missed an infection and it means that they have a reduced cure rate, then that's not so good for the cow. You also might be using it like for culling decisions. So, obviously cows, they're either culled or kept. Will you cull a cow when she could be cured? If you've got a farm that's got lots of replacements coming in, they're probably not too fussed because they've got lots of replacements coming in. Probably a big deal for the cow though. She probably would have liked to stay. If it's a farm who is, short on replacement numbers, if you keep the wrong cow and then they end up infecting other cows, then that's probably a significant thing for them. Buy or buy not. So this is obviously a big issue if you miss a strep ag. And there's also changes in management or infrastructure that's not necessarily needed. So for example, you might, if you misdiagnosed a strep viverus as a problem on farm, you said, oh, we've got to do some teat sealant, we've got to do that, and they didn't actually need it, then that's an investment that potentially they didn't need. Similarly, if you misdiagnose a strep ag and you think, shivers, we've got to treat the whole herd with antibiotics, and that wasn't strep ag, and that's a massive economic thing, so a massive thing that's costing the farm. You might go, oh golly, we've got E. coli, geez, a feed pad, that'll be the best thing for you, a concrete feed pad, and they invest in a concrete feed pad, which I can tell you right now are super expensive, but if it was actually staph, you've wasted their money. So just think about what are the implications of a false negative or false positive test result. You also need to look at the economics. Now, apologies, a webinar is probably not the best place to throw a table like this at you, and because it's actually quite hard to explain and take in quickly. But what I did is I just thought, well, if you're talking about the economics, you want to look at the cost of the actual test. You want to look at the cost of treatments, so the cost of the antibiotics, the cost of the milk down the drain, and then what I've done, so again, apologies, this is hard to explain quickly, but so for example, the cost of on-farm culture might be $30 per cow, but the cost of just treating every case with lactating cow treatment regardless is $108. If so you set something like this up in Excel so that it does the maths for you easily, and you make it so, for example, you can just enter different variables. So you can say, all right, I've got 600 cows here, 2% of the cows have got clinical mastitis. Of those clinical signs, 10% are E. coli or no growths, and then that will give you the number of clinical signs. And then you can say, well, look, if I treat all of them with lactating cow treatment regardless, this is how much it would cost. This is how much it is to test everything, or all the clinical cases. You don't treat one more because it's E. coli, and you save about $130, which means overall, for this particular scenario, you actually negative. It's costing you money to do it. Versus this farm, or this scenario where it's a 600 cows, 10% of clinical cases, half of them are E. coli and no growth, which means, this is what, $6,000 to treat everything, $1,800 to test all the clinical signs. You don't treat 30 cases, which means you save almost $3,000 for not treating, which means overall they're about $900 up. So it really helps to get an Excel spreadsheet up, because it can do the maths more quickly than you. If there's something that I've missed in this one, please let me know, because obviously I want to do this better as well. There was a paper a couple of years ago that modelled the cost effectiveness of on-farm culture and selective lactating cow treatment. And one of the things that they found, so they use a computer program or computer model, the biggest impact on the cost effectiveness was the proportion of gram-positive cases that farm had, and also the difference in likelihood of a cure between blanket and selective lactating cow treatment. So if the chance of the cow curing went down, the more it went down with selective lactating cow treatment, the less economic it was to do. And so, for example, this might be the test itself, or it also might be, like I said a couple of slides ago, whether there's a misidentification. So, for example, if the workers misidentify a strep as a staph or a strep as a E. coli, and they don't treat it, then that means it's not as effective for the farm. And just what they had here was, they say in their conclusion in the paper, on-farm culture should be adopted after careful consideration of the predominant pathogens present in each herd and an honest discussion about the uncertainty surrounding its overall cost effectiveness. So I guess my take home from this is, you don't just say to a farm, hey, you should do this because it's great and be fantastic, but say, look, let's see what pathogens you have, because if you've got lots of no grades and lots of gram negatives, you'll actually be saving money in reductions in antibiotic treatment. And managing people's expectations around setting up a system are also really important. In terms of implementation, so what have we got? Once committed, you need to source a product. Now, a lot of these, actually most of these products, you have to buy in from overseas. So, you actually need an import permit for a lot of these, which is a challenge in itself. You need to have an appropriate setup. So both if you're doing things in clinic and also if you're setting up on farm. So you need to consider your lab requirements. So if you do a Google search will help you come up with the things that you need to fulfill. You need to consider how you're going to collect milk samples and store them properly, how you're going to store the diagnostic tests. Trust me, the Agar plates don't like being frozen. How you're going to label the samples, how you're going to record the results. So whether that's on the farm system or your system at work. You need functioning fridges and incubators. Beer fridges, they're not actually appropriate. They're not going to work for you at keeping things at the temperature they should be. Hygiene, gloves, food, etc. Having a Coke in the fridge that you're storing all your samples in is not appropriate. And you need to consider OH&S implications as well. So I see people are able to use the chat. So over the next couple of slides, I'd love if anyone wants to have a go at writing down what they think we're looking at down the microscope in this picture here. And I'll come to it in a couple of slides. You also need enthusiastic people with the drive and the capacity to put this in place. It's not, someone has to drive to make it happen. And what you also need is whatever system you set up needs to have easy to follow instructions that allow standardized, repeatable results. And this is just a quote from the NMC lab book. It says, microbiological procedures used in the diagnosis of intramammary infections must be accurate. Approximately 90% correlation among interpretations of the same culture by different diagnosticians is expected. So basically, if you have a product and people who all have the same amount of training or some training all look at it and they all come up with different answers, either your instructions need to be better or their training needs to be better. And here's an example of this whole training and interpretation. So SIPCA, they did a paper last year and they compared the results from interpreting the Acumast and the Minnesota bi and triplates with lab culture for different levels of expertise. So they had a trained reader. So he was trained in interpreting these products, and he had experience, or they had experience, and they had moderate to very good agreement with the lab culture. 5 vet students, on the other hand, who all they had was the brochure. So they had the brochure for the Acumask and they had the brochure for the Minnesota triplates. They had poor to good agreement. So they had no training, they were just using the brochure and they didn't perform as well. So the authors say you need to provide training beyond the instruction manual if these are going to work well, or any product is going to work well. The next step of implementation is, if you're going to be doing this on farm, you need a lactating cow treatment protocol. So I've actually taken this slide from a clinical mastitis webinar that Mark and Steph organised a couple of weeks ago, I think, Steph. I'm not quite sure how many people took part who are actually in this meeting, but this is the protocol that they had up on screen. And obviously you just need to modify it to take into account on-farm culture. So for example, for severe mastitis treatment, culturing isn't going to change your treatment protocol, because if a cow's sick, she needs treatment straight away. You'd only be culturing it for interest's sake only. So remember Monica's quote from right at the very start, you only change it if it's going to, you only use a diagnostic test if it's going to change your course of action. So this is only just for interest, really, unless, actually no, I tell a lie. If you've got cows after drying off that are sick, you might say, actually, we do want to know what this is, because if it's Pseudomonas, say, this will change our drying off protocol. So I take that back. You probably do want to culture it to see what it is. So in Mark's protocol, the next step was to look at the third, is this the third case of mastitis in lactation? And then they say, if it is, you look at, talk with the manager about the course of action. And a culture might help a culling decision here. So for example, if you've got someone who, I'm just thinking of a farm in my head where, you know, they like their cows very much. They might give them more than 3 goes for clinical mastitis. But if you can say, actually, it's staph, it is staph, she's had 3 cases, she's never going to get better, she's unlikely to cure with dry cow, it might help that culling decision. And then after that, I've actually modified, this is just an example modification where we've changed it. If there's moderate swelling, heat or tenderness in the udder, you give them supportive treatment while you wait for the milk sample to grow. And if it's gram-positive growth and there's more than 1/4 affected, you go into your moderate mastitis treatment. If there's only 1/4 affected, you go to mild mastitis treatment. And then I've just written here that you don't treat gram-negatives or no growths, and you record all results on the computer. So for any of you who sat in the last webinar and have taken this lactating cow treatment protocol and incorporated it into your own practice, this is suggestions of how you might incorporate rapid mastitis culture into this protocol. I hope that makes sense. Implementation follow-up. Hey, did anyone have any answers as to what this was, what they thought it might be, Steph?
No, Zoe, we didn't get any responses. Sorry, no bites.
Oh, really? No one? Oh, well, hang on, actually. I'll just keep on going this slide for a bit more. So you need, with the follow-up, you need training, you need mentoring, and you need monitoring of progress. So things to consider is, are they reading the results at the right time? Are you reading the results at the right time in your clinic? How are you going to review those? So are they going to store the plates and then once a week you check them to see how they're going? Are they going to take photos so you can have a look to see how they're going and whether they're assessing them right? For the colorimetric systems, are you going to take photos to see whether you're interpreting them right? Same with the Petrifilm. You want to monitor, if you're able to, percentage of contaminated samples, monitor the percentage of no growths, look at the records. Are they matching, the results of the culture matching with the lactating cow treatments? And how are they going to be discarding these products? So this is Nocardia, because it's a gram-positive branching bacteria. Nocardia is, it's probably the one of the mastitis pathogens that is zoonotic. It can cause pneumonia. It actually has the same produces the same substance that tuberculosis does, which, you know, tuberculosis causes nasty pneumonia. So it's not actually something that you want to have. So managing biological substances is actually really important. Know your limits. So for vets, don't use an imaginoscope. If it's contaminated, ditch it. So this is the picture I showed from a few slides ago. Lots and lots of pretty colours. You just need to ditch them because it's contaminated. Don't try and picture something that might be there, but you can't be sure is there. Also remember that we are not microbiologists. I'm quite keen on microbiology, and I think I know way more than I did when I was at uni. But I've talked to microbiologists and emailed them about things I've done, and I can hear them shaking their heads at me, even over email. So remember that we are not microbiologists, but we just need to, if we are keen, we need to make sure that the advice that we provide is valuable and reliable. So if you're keen on milk quality microbiology, consider further study, further training in the area. Read up on it as much as you can. Practice doing things. Learn new like I had to learn how to do gram stains again. So there's things you can do yourself without an actual course, if that makes sense. I just added, so this was the last graph I showed you, I just added to it. So your confidence in a test performance also increases with the more experience and training a person has. So if you've read hundreds and hundreds of your particular test, you will be better at it than someone who's just doing it to start with. So the more of these you look at, the better you will be. Now, Mark, he also asked me, he said, how can you give some quality assurance to the results? And it's a bit hard because how you can do it is you can take a sample that you've done and you can then do it in-house and then you can send it to a lab. You can do that, but it also costs lots of money. And that's money that your clinic has to be prepared to spend, or you have to be prepared to spend, and you might not be prepared to spend that. What I did was I did the basics. I bought some blood agar plates and some McConkey plates, which are really cheap, and I'd plate them at the same time in-house, and I would compare the growth patterns. So these ones here had the same amount of growth, and they were all both gram positive. These ones had they had no growth. So I just looked for the same. I looked at gram stains and growth patterns. I didn't do any extra tests because, like I said, I'm not a trained microbiologist. I didn't have the capacity to do it, but it certainly gave me a little bit of confidence that they were saying pretty much the same thing in terms of gram positive, gram negative, and the amount of growth. And I guess what I would say is because this, how do you quality assure a product without costing lots of money? That's where you want to pick a product that you have confidence in the first place. More about knowing your limits, Mycoplasma and Strep Ag. So I think none of these pick up Mycoplasma except I think the Speedman colour had a spot where Mycoplasma would grow. This is one that's over in Europe, so I don't know how much Mycoplasma it can actually pick up. But a lot of the products still can't pick up between Strep Ag and other strep species. So for example, these are the plates, this beautiful turquoise, it could be Strep Ag or it could be Strep agalactiae. You can't tell the difference. So this is where the PCR is still important. So if we have farms that are growing lots of this light blue, we run order a PCR that's looking for the DNA of the different bacteria. And we have a look and we go, oh, fantastic. There is no Strep Ag. We just have Strep agalactiae. Of course, obviously, you also look to see whether there's Mycoplasma. In this case, there was Mycoplasma species. So, don't forget about looking for Mycoplasma, especially if you have samples with lots of no growths, and especially if you're purchasing cattle. There is lots of, the cases of Mycoplasma that I've seen recently or where it's come up, it's all been when animals have been purchased and have changed farm. Farmers are also not microbiologists, and again, it's this looks clean, but it's still got still got 3 types of bacteria, so it's classed as contaminated. So it's important that they are clear on how they interpret plates. Also, there'll be some biases that farmers will have that we don't. So for example, oh, it's Betsy, I'll give her some lactating cow treatment, or I'll keep her anyway. She's been, even though she's had 10 cases of clinical mastitis this year. So they have some biases that we don't. And then also, is it going to be an added complication? So, you know, if you suspect that it's a farm where misinterpretation of the test might be an issue, or whether poor hygiene will contribute to unreliable results, then it's probably, or if there's lots of other stuff going on apart from milk quality that you just suspect they're not going to be able to do this well, then maybe they shouldn't do it on farm, and take the decision and the interpretation part away from them. So they still take samples, but you might run the tests in-house, or it might be that you send it away to a lab. And I think this is particularly important if they do actually have a mastitis problem, because if you're investigating a mastitis problem, you want to make sure that you give them the best results, or most accurate results that you can. Now, what about antibiotic sensitivities? So a lot of these tests, they don't actually have antibiotic sensitivities, and you're not sending them away to the lab. Hey, I've got another question for the chat if anyone's up for it. This is a very cool old antibiotic susceptibility disc I found in the clinic a couple months back. My question to you, who is older, this or me? So we'll see if anyone has any answers at the end of it, Steph. So anyway, do you need antibiotic sensitivities? So I guess working from first principles, I guess you look at what bugs are out there and what products are out there. So this is how I look at bugs. I have lots of fluffy bugs that I get from giantmicrobes.com, and I use them in my milk quality presentations. I find it good to have something visual for people to look at when I'm talking about Pseudomonas and talking about staph. And so this is how I group them. So I group this lot over here as the bugs where antibiotics aren't going to do any good whatsoever. So your Prototheca and Serratia and Nocardia and Pseudomonas and Mycoplasma, doesn't matter what you use because it's not going to work anyway. Our next group is our coliforms, so E. coli and Klebsiella. So they're actually in our no treat category anyway. So the idea is that you don't give them any antibiotics, but if you do know you have those, well, you don't use your penicillins. Staph aureus I've got here on its own because antibiotics aren't always of use, only because it's got lots of other things that help it fight back against the cow. And so what we've got left is gram-positive bacteria, so C. bovis and streps, coagulase negative staph, and in theory, all the products available in Australia should work. They're all designed to work against the bugs that they're going to. So I guess what do you do? If all of the products that should work, like, how do you choose? And this is just another quote from NMC, and that says, susceptibility testing is not very useful in determining the outcome of treating an intramammary infection with an antimicrobial. Empirical evidence of what works for a given organism on the given farm is the usual approach. So it's kind of, is this product working for you? Yes. Well, then keep on using it, I guess, is the approach. What I find is it's not just preferences, it's also logistics that helps choose which product you're going to use. So for example, if you've got a farm where they only treat once a day because it's only the manager that does the treatment and he only does it in the afternoon, then you're limited to only 2 products or 3 products. It might be a farm where they're all treatments are given intramuscular because their intramammary hygiene is poor. And I remember, I used this example from John House. Sorry, John, if you're there, but he described a farm with Mycoplasma where he sat there and watched as they were stripping cows and the little droplets of aerosols drifted from an infected cow onto the lactating cow treatment sitting on the platform next to it. So, you know, it might be, doesn't matter which intramammary you might use because you're giving them intramuscular. You might have a farm and they just really want a short milk withholding period, which limits the product that you can use. They might want a short meat withhold. Again, there's only a certain number of products that have short milk withholds. Or they might just want the longest time in the udder because they want to maximise, you know, the time that the antibiotics are there. So this is how I find farmers seem to choose their products more, you know, what works for their farm. And I guess this last slide is just, I guess you'd call it my soapbox. See, cow's milk soap. And I think it's really important that rather than having that focus all on about antibiotic treatments, is to shift that focus of farmers onto prevention. And I think it's really important that we learn about the bacteria and the virulence factors they have, because just because something works in the lab doesn't mean it's going to work in the cow, because in the cow, the bacteria are able to fight back. So for example, they have capsules which stop the antibiotics from working. They can adhere and form biofilms. They are motile, like Pseudomonas. They have the lipopolysaccharide that, you know, makes the cow's immune system go crazy. They have enzymes that destroy the white blood cells. They have the enzymes that destroy the tissue cells. So, you know, this is why staph's hard to treat, because it damages the tissue and gets into cells. And it doesn't matter how amazing the antibiotic is if it doesn't get to it. They have siderophores that capture iron. They can inhibit complement and the antibodies, and they can camouflage themselves from the immune system, and they have oxygen radicals. So these are so many virulence factors that all the different bacteria can have, and that's not even counting, you know, the antimicrobial resistance that some bacteria have, you know, just naturally. So I think it's really important, if you're keen on microbiology, to learn about these and start talking with our farmers about, geez, you know what, you know, these bacteria, they fight back. It doesn't matter about antibiotics because they can still fight back. Let's prevent the infections in the first place, and really focus on a happy and healthy immune system so that the cow can, you know, fight the bacteria as well as she can as well. So in summary, I think we've made it in terms of time. You want to know your reason for wanting a diagnosis. Is it a mastitis investigation for selective lactating cow treatment, culling decisions, biosecurity? You want to choose products with published peer-reviewed research. You want to consider the ease of use of the product and the clearness of the instructions. You want to consider the capability, and some farms and or some vet clinics are just not going to be suitable for rapid treatments, rapid diagnostics. You want to consider the economics of it. So the farmer desperately want to do it, but if he only has strep mastitis, then will it actually be worth it? And you want to consider how you will monitor, retrain and upskill staff. And Steph, I can stop talking. Are there questions?
Thanks, Zoe. That was fantastic. If anyone's got any questions, if they could just throw them into the chat box. So just as a reminder, if you just hover at the bottom of your screen, there's the option for the chat. Otherwise, if you're on a smartphone, just tap your screen and the 3 dots on the bottom right-hand side. So Zoe, while I'm waiting for questions to come through in the chat box, we had a question from Werner during the presentation on the economics of treatment. So it says, you might be saving $900 if you're not treating the cows now, but will this cost you money in the long term?
Oh yeah, so I guess if you're not treating, say, coliforms or no growths, so the studies went, like those first 2 studies, I think they were, I can't remember which slide they were. It basically said when they've done this, there wasn't any difference in this particular study in cure rates or recurrent clinical cases. So for those, you know, for that study, it didn't cost any money in the long run because you know, the cure rates and the weren't different. But if you had a system where, so for example, they didn't diagnose the cases properly, or they had just a different, like, so I'm just trying to make sense here. If they misdiagnosed or what have you, then those results might be different. So, so in theory, all the studies have said that when you do selective treatment, it hasn't made a difference to cure rates. But these are studies done by people who are better trained than our average farmer and have probably there's a bit more of an investment in making sure it's all going right. So if you have someone who is just let loose without any mentoring or guidance, then it might actually cost them. Does that make sense, I hope? Kind of depends on the quality of how well you implement the system.
I think so, Zoe, and I think it also probably really highlights the importance of having confidence in the testing that you use. So you mentioned sort of around who is actually doing the testing, but also making sure that using a test methodology that you're comfortable with its accuracy, etc.
Yes.
So yes, you've answered that very well. A question from Peter Eunice. Zoe, can you recall the correlation between the labs in the Ferreira paper? I think that was the Acumestin Minnesota Bi and Triplates paper.
Well, no. So with those papers, what I've just done is I kind of summarised for each, like for all the 4 products. I've just done the summary. For this presentation, I've just summarised the kappa because that was the easiest way to compare it on a slide. So I do have the papers, and which have all the extra specificities and sensitivities. I just can't remember them off the top of my head. I need to have them in front of me because there's just too many numbers.
Okay,
sorry, Pete. Yeah, I can email it to you, Pete. But, yeah.
Yeah, that sounds great. Okay, next question from Marg. Have you got any comments on milk-based antibody testing?
Any particular type of antibody?
Marg, yeah, she hasn't said, but perhaps, oh, sorry, no, I can see, referring to Staph aureus antibody testing in milk.
So are you testing the antibodies in the milk or antibodies in the blood? Must be antibodies in the milk. I don't know. Sorry, I haven't.
No, that's okay.
Yeah, so it's yeah. Were you going to say it's Mark Humphreys here, Steph? That's certainly being started in New Zealand through the herd test centres, and we've had some discussions with them, but still reasonably early stages in terms of the, I suppose, again, the predictability and accuracy of that just at the moment.
Yeah, I guess what I would say to that is it's a bit similar to PCR. It tells you that the bacteria was present, but it doesn't tell you if it's active. So if I'm a cow and I've been exposed to staph and I've mounted an immune response and I got rid of it, I'll have antibodies, but I might not have the bacteria. And that's the same with PCR. If I do a PCR of the milk, I'm like, oh golly, there's staph there. It doesn't tell me whether it's alive or dead. Where I think, and this is what I personally think, is for something like Mycoplasma, it's probably more useful because you can say, like with Mycoplasma, it doesn't matter if it's an active infection or not. You want to know it's there, and Strep Ag as well, because that's how we use PCR. You know, it's not about whether there's a bacteria that's alive. You're just looking for its presence in the first place. Because if you've got Mycoplasma present, how you approach that cow or the farm that you're going to buy it from is completely different. Does that make sense?
Yeah, that's great. Thanks, Zoe. So I'm just conscious that we are now running over time. So what I think we might do, Zoe, are you able to just flick to the feedback slide? And
yes, this one.
Yeah, so I guess the call out, so what I'm going to do is we might take a couple of more questions, Zoe, if you're happy to do that. So we might go to about 10 past, but if anyone does have to run, if you can scan the QR code that is currently on the screen with your phone, that'll bring up a very quick feedback survey. Really, we just want to know what you think. But most importantly, do you want more webinars? Has this been useful? And if so, are there other Dairy Australia topics that you'd like us to cover? So we'd really appreciate any feedback. We do use it, and that was the impetus for this webinar. So if anyone's got to run, if you could fill that out. It takes 2 minutes before you run.
Hey, Steph, can I say, I would like Dairy Australia to organise a webinar that will teach my children at school at home, so that I don't have to do it for an hour. Could you please organise that?
I'll see what I can do, Zoe.
That would be great.
Cool. All right. So the next question is from Joel H. So Zoe, do you have any thoughts on selective therapy based on individual cell counts instead of culture? Now, you haven't mentioned, Joel, whether that's, I'm assuming that's related to selective dry cow therapy. Not sure. Perhaps if you can come back in the chat box and just confirm whether that's what you're talking about. Stuart, just while Joel's coming back to us, Stuart's asking, hi Zoe, are the tests profitable for the clinic, and how does the price for a farmer compare with lab testing?
So I guess I can't speak for other clinics, I can only speak for our clinic, but if you send sample away to the lab at the moment, I think you're looking at about $85. And that, so there's, you know, there's a dispatch fee, there's a lab fee. I don't know if that includes the sensitivity or not. So you're looking at $80, $90. And it used to be that the more samples you sent, the cheaper it was per sample. But that's actually changed for our lab, where now it's just the set price regardless of the number of samples. So we've made it a fair amount cheaper. So you could probably get 4 tests for the same price. And I guess this is probably partly my drive, because I want more farmers to do the testing, because then we can give them better answers. I guess, like, if you've got a problem, and it's got to be, like, if someone, I guess partly this is also my dairy farmer hat on, is I want it to be something that's affordable and quick, and we make money. I don't know whether it's stacks and stacks of money, but it's, yeah, it probably makes a bit of money, not stacks and stacks of money, but it's certainly better for the farmer, and it's quicker for the farmer. And since we've started doing it, we do a lot more mastitis testing.
Yeah, thanks, Zoe.
That would be, sum it up.
No, that's great. And it actually probably leads on to the next question, if you're happy to keep going. So Pete Eunice would also like to know, can you comment on the growth patterns of a sample that was plated up within a few hours compared to one that you might send via a courier to a lab that's not plated up for, say, a day or two later?
I guess this is, yeah, what I would say is that I probably look at these more than I should. I should probably clean my house, but I like to take photos of these plates. And what I'll find is that the first day you'll have pure growths, you know, really obvious, you know, one particular bug, and then maybe 2 or 3 days later, like, these other little random ones will kind of start to appear. So I take these are like the bugs that are probably normally in the milk, because milk's not necessarily sterile, and they've just taken some time to grow. So what I imagine is, if you've had something sitting there and then it takes, you know, a day and a half to get to the lab, and then by the time they plate it, you might be more likely to have, like, something that's classed as contaminated. And certainly with the studies that looked at the rapid mastitis testing, apart from one study, they were all done later. So they'd all been frozen, then sent to the lab and done differently. So that will change some of the, there will be a difference, I think, on results. But if you can just make sure that you get it to the lab as quickly as possible, collect it as hygienically as possible in the first place. So for, you know, yeah, so that there's minimal chance of any contaminants overgrowing, that would be best. I think, yeah. That kind of answered the question, I think, Pete. But what I would say is like I said, I like to look at things, and watching contaminants grow is truly fascinating. You know, I sometimes plate some calf milk, you know, calf waste milk up on these things just to see what they look like and what we're growing. Amazing what's in that milk.
Yeah, absolutely. So just a comment here from Joel Dutton. So Joel says that implementing a treatment protocol is a lot cheaper than investing in a rapid testing system on farm. So yeah, really valuable comment there, particularly if they don't have a good treatment protocol in place currently. I think, yeah, it's a pretty good call out.
Oh yeah, and what I would say, Joel, is focus on detection, the right detection and the right definitions of clinical mastitis, and hygienic treatment and storage of products, so that they're not actually, you know, so that the products are clean and the ends of the teats are clean when you infuse them. And every so often I'll have a look at some mastitis focus reports, and I'll go, oh, geez, the new infection rate's actually pretty high. And then I'll talk about just a couple of things that are going on farm, and I think maybe the hygiene in the dairy is what's actually causing the new infections. So, you know, focus on things that are really obvious to start with, for sure.
Yeah, great. All right, so we're at 2:09, so I think we might wrap it up. We have still got a couple of questions, and just to those people with questions that we've missed, Zoe, would it be okay if I sent you through these questions and perhaps we get back to these people directly? Great, okay, excellent. So we won't miss anyone. So just before we let everyone go, I just wanted to say a huge thank you to Zoe and to Mark Humphreys, who, apologies Mark, I missed you in the introductions at the start. Just to thank you both very much for all the time that you've put into preparing the webinar for today. It's a pretty massive job for an hour slot, so we really appreciate it. Thank you both very much.
Yep, no worries. I could talk about this all day, Steph. So.
Absolutely. And just to reiterate, the bacterial identification FAQ, which summarises a lot of the material that was covered in the webinar, will be available in the next couple of weeks. And I'll send that out directly to everybody that registered for the webinar, but it will also be available on the Dairy Australia website. We are, just as a final note, we are looking to run a farmer webinar on both clinical mastitis management and mastitis diagnostics, rapid diagnostics, in the week of the 11th of May. So I will send out that invitation to all of you that have registered here, but it may be worthwhile jumping online just to make sure you know, that you're sort of across what's covered in the webinar, that will be presented directly to farmers. So yep, without further ado, I'll let everyone get on with the rest of their day. And again, I hope...
Just as an aside, I'm actually older than those discs.
Oh, good.
They were from 1986 and I'm older than that. So there you go. But thank you everyone for saying that I was younger than them.
Great. Thanks, Zoe. And yeah, we, as I said, can't emphasize enough, if you can just let us know what you think and if you want more. And we'll talk to you next time.
Okay. Thanks, Steph.